Okay. We have as a guest today Professor Alexei Tepikin, who is a well-known scientist in the field of calcium signaling research and research in pancreatic acinar cells. Alexei was born in Kiev, actually, and graduated from school 145 about more than 30 years ago. After that, he entered Moscow Institute of Physics and Technology, and after graduation, he arrived to Kiev, and for about 7-8 years, he studied at our institute, Bogomolets Institute of Physiology, as a student. After that, as PhD student, he defended his PhD thesis, which was devoted to actually calcium signaling in neurons of helix spermatia, which means that his initial stages in his career were devoted to neuroscience, the field in which most of us work now. But fortunately or unfortunately, after that, he moved to Liverpool, to Department of Physiology, University of Liverpool, where he started to study calcium signaling and regulation in pancreatic acinar cells, and the most part of his career has been devoted to this issue, and he continues to do this research now. He switched a lot, because not only calcium regulation, but different stages of, in my opinion, calcium signaling or calcium dependent signaling have been in the center of his research. Now he is for many years a professor in the Department of Cellular and Molecular Physiology at the same university, the University of Liverpool. And today's lecture will be devoted to some interrelationship between calcium signaling, bioenergetics, and autophagy, and everything will be in the field of pancreatic acinar cell research. Alexei, the field is yours. Please start your lecture.
Okay, Pasha, thank you very much for this kind introduction. So we'll talk about calcium signaling. Can you see this? So we'll talk about calcium signaling, bioenergetics, and autophagy, and it will be a lesson from pancreatic acinar cell. So the first slide shows the old building of the Department of Physiology in the University of Liverpool, in customary for Liverpool weather, strong wind, as you can see on these trees. So the building was used by Charles Sherrington. So this is the place where the first electroencephalogram was taken by Ca2. This is the place where Sherrington created the word synapse. This is the place where the cradle of neuroscience was, the cradle of modern electrophysiological neuroscience was established. So this is plan of my today's lecture. I will talk about calcium signaling and distribution of cellular organelles in pancreatic acinar cells. After that, I will move from calcium signaling to mitochondrial function, regulation of bioenergetics. We will talk about store-operated calcium entry in these cells, STEAM, RAI, and the platforms for store-operated calcium entry, junctions between endoplasmic reticulum and plasma membrane. And after that, I will give example of what is known as calcium toxicity. So as you know, calcium is very useful. It is important second messenger, but it is also potentially toxic ion. It can kill cardiomyocytes, neurons. It can also kill and damage acinar cells. And I will talk about one of the manifestations of this calcium toxicity, formation of endocytic vacuoles and intravacuolar trypsinogen activation in these organelles. And at the end of the lecture, I will talk about our recent research in our laboratory about rupture, exocytosis, actination, and non-canonical autophagy of these organelles. So pancreatic acinar cells secrete digestive enzymes. They also secrete fluid, as we discuss later in the lecture. And important secretagogues. Secretagogues are substances which stimulate secretion, and this can be acetylcholine, or it can be hormone cholecystokinin. Utilize calcium signaling cascade to trigger and regulate secretion from these cell types. So this slide shows two isolated pancreatic acinar cells. And this is just one of the types of calcium signals in these pancreatic acinar cells. It originates in apical part of the cell, so this part of the cell actually contains secretory granules. And after that, it propagates as a wave and engulf all the cytosol of the cells. This is another form of calcium signal, so this is important, physiologically relevant form of calcium signals, something which will be manifested during relatively mild physiological stimulation of these cells. So again, these are two isolated pancreatic acinar cells. These dark speckles are actually secretory granules with digestive enzymes and precursors of digestive enzymes. This is something which will be released from the pancreas, will go through the system of pancreatic ducts, will go to duodenum, to intestine, will aid digestion. And you can see that when we stimulate these cells, in this case we stimulate with acetylcholine, we have activation of calcium signals, and these are local calcium signals. You see that they are originated in the area of secretory granules, exactly where they are needed. And after that, this calcium signal matures and dissipates in this area without spreading into the rest of the cytosol. So we studied both physiology and pathophysiology of these signals, and this is one of the techniques which we use to study physiology of these signals, and this is known as calcium uncaging. So we use caged compounds, and caged compounds are molecules which are inactivated by caging groups but can potentially release biologically active substances when they are activated by photons. In this case, we are using caged calcium and called NPGTA. And when we apply pulse of light, apply it again, we can generate these local calcium signals. So this technique, which utilizes caging compounds and uncaging, can be used to generate second messengers locally, as we can see for the calcium. It can be also used to produce reactive oxygen species, to change intracellular pH, to trigger local generation of the hormones and neurotransmitters. In this case, we used it to produce local calcium responses, and after that, we basically combine this technique with electrophysiology to look at the consequences of these local calcium transients. And you can see when we uncage in basal part of the cell, we have large calcium responses. We are recording electrophysiology of these cells, and there is basically nothing happening. In this particular case, we are doing this experiment to look at activation of calcium-dependent chloride currents and localization of calcium-dependent chloride currents. So this is important because pancreatic acinar cells secrete digestive enzymes from this part of the cell, but it also needs to kind of secrete fluid, so the digestive enzymes can be flown, can be conveyed through the system of pancreatic ducts into the duodenum. To secrete fluid, cells need to open chloride channels, and after that, sodium will kind of move paracellularly by osmosis, and as a result, we can generate production of the fluid. So when we release calcium in basal part of the cell, we have calcium responses and no changes of the current. When we release in the lateral part of the cell, we have calcium responses and no changes of the current. But when we can generate this local calcium response, specifically in the apical part of the cell, you can see that calcium transients are accompanied with opening of the calcium-dependent chloride channels. So combining electrophysiology, you can just see the shadow of patch pipette with localized calcium uncaging allows us to identify the position of the calcium-dependent chloride currents, and they are located specifically in the apical part of pancreatic acinar cells. So this is basically just application of the uncaging technique and localization of the ionic conductances in pancreatic acinar cell. So let's return to these local calcium signals. So if their origination in apical part of the cell is well correlated with the presence of IP3 receptors in this region. So this is distribution of IP3 receptors, so all three types are expressed in this cell, so from 1 to 3. It is considered that type 2 and type 3 are particularly important for the secretion in this cell type. This is their localization. So these are clusters of pancreatic acinar cells, and the apical parts of the cells in this area. This is the area where they secrete digestive enzymes and fluid. And you can see that they are basically aligned with the places where we have seen these localized calcium responses. But this localized calcium response is actually a very strange phenomenon. The cell is only 20 micrometers in diameter. So it is a kind of smallish cell, substantially smaller than, of course, neurons can be. It is much smaller than cardiomyocyte usually. But yet, this tiny little cell can generate localized calcium response, which can last for tens of seconds. This is a very, very kind of strange phenomenon. It is kind of, these cells have high calcium binding capacity. So approximately 2000, meaning that kind of there is a very strong calcium buffering. But even this very strong calcium buffering cannot explain this extended, prolonged calcium signal. And in reality, what we need an organelle, which will be able to shape these localized calcium responses. So for a very few kind of minutes, we will now disconnect from calcium signals and look at the distribution of cellular organelles in pancreatic acinar cells. So transmitted light images shows you isolated doublets of pancreatic acinar cells, and you can see that apical part of the cell is occupied by secretory granules. There is Golgi apparatus, which is surrounding this secretory granules. And after that, we have strands of endoplasmic reticulum. This is actually, cell is densely packed with endoplasmic reticulum. It is really protein secreting machinery, so it is highest protein secreting rate among the cells of the body. So approximately 10% of the total protein is secreted and changed every day. So this is powerful protein secretory machinery, so very well developed endoplasmic reticulum lens in the basal part of the cell, with the strands of endoplasmic reticulum projecting into the apical part of the cell. And there is also mitochondria. They occupy approximately 10% of the cell volume, and they are kind of enveloping Golgi apparatus from the basal and lateral parts of the cell. So nice, structured, layered arrangement. Secretory granule, Golgi apparatus, mitochondrial, endoplasmic reticulum. So this is a separate image of localization of mitochondria in pancreatic acinar cells. You can see the distribution of secretory granules, and you can see the distribution of mitochondria. So there is a densest group of mitochondria. We term it perigranular mitochondria or mitochondrial belt, which surround secretory granules of the cell. There are also mitochondria immediately under the plasma membrane of the cell, but there is an important group. This is perigranular mitochondria. So to cut long story short, these are the mitochondria which shape localized calcium signals, which prevent their spreading into the rest of the cell. So kind of term which is mitochondrial firewall, introduced by one of our colleagues in the field, David Hill, but we like this term and we use it extensively. So the principle is that This is the localized calcium signal, and this is the mitochondrial calcium belt, and these are the mitochondria which help to shape these localized calcium responses. So we are now kind of moving to the next part of the lecture. So from calcium signaling to bioenergetics of cellular organelles, or we basically just try to think and contemplate what actually mitochondria do with these calcium signals. Some of these calcium signals actually enters into the mitochondria. So this is again calcium-dependent chloride currents. So the negative deflection of this trace corresponds to actual increase of cytosolic calcium concentration. And you can see that increase of cytosolic calcium concentration is accompanied by increase of mitochondrial calcium response. In this case, we are measuring mitochondrial calcium responses using Rod2. This is calcium indicator, but it has delocalized positive charge. So the localized positive charge is hidden from the lipid. Indicator can actually load organelles which have negative voltage in comparison with the cytosol of the cell. Of course, you know that mitochondria have negative voltage of approximately minus 140, minus 160 millivolts with respect to the cytosol. So if you stick an electrode into mitochondria and cytosol, you will see it basically will show the negative voltage of approximately minus 140, minus 160 millivolts. So positive charges are attracted to mitochondria matrix. This allows us to preferentially monitor mitochondrial calcium responses using this indicator. So you can see that cytosolic calcium responses are accompanied by changes of calcium in mitochondria. When these responses are relatively small, it is mitochondria, which are mitochondria preferentially around the mitochondria belt, which takes up this calcium. So this is the mitochondria around the secretory granules. When we apply a very strong supramaximal stimulation, in this case with acetylcholine, then all mitochondria of the cell respond. These are not calcium oscillations, these are actually oscillations of the rate of the Krebs cycle. And we can measure this very important bioenergetics parameter by monitoring autofluorescence of the cell. And this autofluorescence of the cell, in this particular case, it is autofluorescence of an ADH. So an ADH is, of course, as you know, produced in the Krebs cycle. This is important reducing equivalent. Reducing equivalent is donor of electrons. So this important equivalent is produced in the Krebs cycle, and utilized as a donor of electrons in electron transport chain. Fortunately for us and other people working in bioenergetics, this molecule is fluorescent. So you don't need to load cells with anything, just sit in darkness, illuminate them with approximately 360 nanometers light, and it will show you the changes of an ADH concentration in the cell, preferentially in mitochondria. This is how it looks like. So these are oscillations. Each of the spike is changes of the rate of the Krebs cycles. So these are oscillations of the rate of the Krebs cycles in this cell. The lower part of the slide shows distribution of an ADH. This is specific mitochondrial staining, one of mitotrackers. And you can see almost perfect colocalization of the two staining, indicating that we are indeed measuring mitochondrial responses. So as I mentioned, each of these transient is actually consequence of calcium changes. So again, this is calcium-dependent chloride current. You can see that cytosolic calcium response is accompanied by changes of wave of NADH production. So this is actually illustrates an important phenomenon known as stimulus-metabolism coupling. So you are now familiar with stimulus secretion coupling, with stimulus contraction coupling. This is an example of stimulus metabolism coupling. So this is the link between initial stimulus, hormone or neurotransmitter, second messenger, and adjustment of metabolism to the demands of this particular stimulus. Cell is anticipating that calcium signal is already generated. Cell is preparing to secrete. Cell is preparing to secrete digestive enzymes by exocytosis. This is energy-dependent process. Cell is preparing to secrete fluid by opening of chloride channels, by changing ionic gradients, which will need to be restored. So these are all energy-consuming processes. Cell anticipates this and upregulates the rate of its Krebs cycle to meet the increased energy demand. Stimulus metabolism coupling. So we recently had opportunity to look at this again, and we had this opportunity because of the generation of the mice which are deficient on mitochondrial calcium uniporter. So mitochondrial calcium uniporter complex is a mechanism, preferential mechanism for mitochondrial entry, for calcium entry into the mitochondria. We also have changed our way of measuring mitochondrial calcium. In this case, we are using mitochondria targeted probe, mitochondria targeted chameleon. And this is how cells look like. So again, we can see this preferential localization of the indicator around the secretory granular areas of the small cluster of the cells. This is our mitochondrial belt. This is how the knockout animals look like. So mitochondrial calcium uniporter is completely eliminated in these cells. And this is what is the kind of physiology of these calcium responses in mitochondria look like. This is wild type cells when we stimulate them with calcium releasing agonist. In this particular case, it is cholecystokinin. There is large calcium entry into mitochondria of the cells. In MCU knockout mouse, calcium entry is largely, mainly suppressed. So this is another inducer of acute pancreatitis. This is actually bile acids. So biles are among the known inducers of acute pancreatitis in a very particular conditions, when gallstone actually blocks the ampulla of water. So in this case, there is a possibility for bile to reflux into the pancreas. And this is considered as one of the possible mechanisms of acute pancreatitis. So bile acid, in this particular case, it is TLCS, taurolysopalate, can trigger also cytosolic calcium responses. And in wild type cells, this is accomplished by mitochondrial calcium entry. And in the knockout animals, this mitochondrial calcium entry is mainly eliminated. And we had the opportunity to look again at these NADH responses. So this is changes of the rate of the Krebs cycle, as I mentioned, in condition when we knocked out mitochondrial calcium uniporter. In these animals, you can see that the response of an ADH to this calcium releasing agonist is substantially reduced. There is another fluorescent molecules. This is FAD. In this case, it is the oxidized form of FAD, which is fluorescent. It is actually utilized in the Krebs cycle, so its decrease is corresponding to increase of an ADH. So we can see that there is decrease of the FAD response also. So this is the ratio, and again, the ratio indicates the changes of the rate of the Krebs cycle. And you can see that knockout of mitochondrial calcium uniporter drastically, albeit incompletely, suppresses the stimulus metabolism coupling in mitochondria of these cells. So it is conceivable that there is still a tiny little calcium influx into the mitochondria by unknown to us mechanisms. So this is something which is rather intriguing. We don't know what is this, so we can speculate at the end of the lecture, discuss this. Nevertheless, you can see that there is significant reduction in the rate of the Krebs cycle in the condition when we knocked down mitochondrial calcium uniport. So finally, we can actually measure ATP concentration in the cytosol of pancreatic acinar cells. And we can do it using luciferase, which delivered in these cells via the replication deficient adenovirus. So this is primary cells. We express it using adenoviruses. This is calibration protocol. When we increase ATP concentration and permeabilize the cells gently with alpha toxin, so proteins will not go through the alpha toxin pores, but ATP will, and increase ATP concentration outside, you can see that the brightness of the cells will increase. When we use millimolar concentration of ATP, brightness don't change very much. When we remove ATP and permeabilize our cells, ATP of course leaks from the cells and they become dimmer. So using luciferase, we can monitor ATP levels. We can monitor it in cytosol of the cell. We can monitor it in mitochondria of the cells. And you can see that when we stimulate the cell with calcium releasing agonist, ATP concentration in the cytosol actually increases. And this is the kind of triumph of stimulus metabolism coupling in this cell type. So cell increases energy consumption. Oxygen consumption increases 3 times. ATP usage increases something like 3 times. And yet ATP content of the cytosol actually increases. And this is basically the result of this very efficient stimulus metabolism coupling in the cells. This is a result of calcium entry into the mitochondria, upregulation of the rate of the Krebs cycle, and upregulation of the ATP, mitochondrial ATP generation. There is interesting feedback. So calcium has an important effect on the metabolism, and there is also feedback from metabolism to calcium signaling. So this metabolism calcium feedback is manifested by the phenomenon of the no ATP, no signaling. So when we inhibit mitochondria, so it is very effectively blocks calcium signaling in this cell type. So cell detect that cell is losing energy, calcium signaling energy consuming, and therefore it very effectively drops calcium signaling in the cell. So this is ATP measured in the cytosol of the cell. This is inhibition of the both mitochondria energy production and glycolysis. Oligomycin blocks mitochondrial ATP synthase. Iodoacetate blocks glycolysis. So ATP drops, and kind of relatively modest decrease of ATP blocks like a knife into hot butter, blocks generation of calcium signals in this cell type. So stimulus metabolism feedback. So this is the principle. No ATP, no signaling. Decrease of ATP content inhibits calcium oscillations. It also inhibits calcium leak from the endoplasmic reticulum. It inhibits calcium releases from the IP3 receptors. IP3 receptors are ATP sensitive, particularly apparently IP3 receptor type 2. And it inhibits store-operated calcium entry. Okay, so far we were actually mainly talking about kind of useful things which calcium can do in the cells. It can stimulate secretion, it can stimulate secretion of fluid by opening chloride channels, it can upregulate ATP generation in mitochondria of these cells. But long-term calcium signals, they are of course global calcium signals, and they are not only global, they are actually long-lasting. They can actually damage pancreatic acinar cells. So this long plateau of the cell is actually, calcium increase is actually important for this damaging effect, for this calcium toxicity. As you probably already know, that this calcium entry is mediated by store-operated mechanism. I'll return to this transient. So this part is the release from intracellular stores, but of course the calcium content of intracellular stores is limited. Without calcium entry, calcium pumps of the plasma membrane will basically just run it down very quickly. So to form the plateau, we need continuous calcium entry into the cell. In this cell type, there is no voltage gated channels. So the main mechanism is calcium entry via store-operated calcium channels. And there is a very peculiar, very strange, and already known to you mechanism, probably. I heard that you already had a lecture about 24I, is mechanism which is based on the interaction of the two proteins which belong to different organelles. So STIM, which monitors calcium content in the plasmic reticulum, in lumen of endoplasmic reticulum, and ORI, which is channel-forming component of this pathway localized in the plasma membrane of the cell. And interaction of the two proteins which belong to different organelles results in the store-operated calcium entry. It can happen only in very specific part of the cell. It can happen in the junctions between the endoplasmic reticulum, plasma membrane of the cells. This is how these junctions look like in pancreatic acinar cells. So these are two pancreatic acinar cells. They are expressing fluorescently labeled STIM molecules, and upon depletion, upon stimulation, release of calcium from the endoplasmic reticulum, it translocates to the junctions between the endoplasmic reticulum and plasma membrane of the cell. So these little stars are actually junctions between the endoplasmic reticulum and plasma membrane of the cell, junctions decorated with fluorescently labeled STIM. So the translocation of the STIM is aligned with the translocation of ORI. So this cell is expressing STIM, stimulated, translocate to the plasma membrane, forming this little punctum. This is ORI. Upon stimulation, translocate to the plasma membrane, forming puncta. And this yellow color shows the colocalization of the STIM and ORI in the plasma membrane of the cell. So in this study, Georg Gelur first identified STIM1 and ORI1 as mediators of this pathway in pancreatic acinar cells. And later, Alyosha Grishenko actually characterized electrophysiology of these channels. So we looked a little bit more details of the this platform for the signaling. So this is the junction in which STIM can interact with ORI and trigger the opening of the ORI and store-operated calcium entry. Junctional complexes. You can see the strand of endoplasmic reticulum studded with ribosomes. Suddenly ribosomes disappear and it form contacts with the plasma membrane. So another example, you can see a strand of ER coated with the ribosomes all the way, approaches plasma membrane of the cell, and you can see the two membranes basically counterfollow one another at some distance, and this is the places where STIM can interact with ORI and trigger the influx of the calcium into the cell. So this is basically how this plateau is formed. So this is what it is capable of doing. So these are three isolated pancreatic acinar cells. We are stimulating them, but we are now stimulating them in a condition which is damaging to the cells. So we are stimulating with high doses of the cholecystokinin. This is something which is used in experimental acute pancreatitis to trigger acute pancreatitis. This is used in cellular models of acute pancreatitis, and then we use this high concentration of the agonist. What we see is formation of these large organelles, and these organelles are endocytic vacuoles. This is example of the confocal microscopy in reverse. In this case, we do not put any indicator in our cells, and therefore cells form dark shadows on our confocal section. So basically just nothing to fluoresce, dark cytosol. But cells are placed in brightly fluorescent solution which contains dextran. So this is dextran with fluorescent marker. And therefore when cells endocytose this dextran, and it's kind of large molecules, they can't really move through the plasma membrane of the cell at molecular weight from 3 to 10,000. And these dextrans are labeled with fluorescent indicators, and when cell endocytoses, and this is the only way for the molecules to enter the cell by endocytosis. So what we actually reveal is endocytic structures. So these are endocytic structures, endocytic vacuoles. Diameter of the cell is 10 micrometers. These endocytic structures can be up to, the diameter of the cell is 20 micrometers. These endocytic structures, endocytic vacuoles, can be up to 10 micrometers in diameter. They originate in apical part of the cell, and after that move all the way up to the basal part of the cell. So you remember this nice layered arrangement of cellular organelles in pancreatic acinar cells, secretory granules, Golgi apparatus, layer of mitochondria, endoplasmic reticulum, and suddenly these cannonballs, which are 10 micrometers of diameter, starts to throw secretory granules, punch through the mitochondrial belt, and to crush through the layers of endoplasmic reticulum all the way to the plasma membrane of the cell, a real chaos in the cytosol of the cell. This is how these endocytic vacuoles look like in electron microscopy. And this is the result of the aberrant compound endocytosis. So normally only few secretory granules fuse, up to maybe 3 secretory granules. In the process is known as compound exocytosis. So first secretory granule fuse with the plasma membrane of the cell, and after that the second and third fuse with the first secretory granules. This is very economical way. They don't need a lot of space. They basically can secrete through this compound exocytotic structure. But when very high concentration of calcium is generated by this very large concentration of secretagogue, then instead of 1-2 secretory granules, something like 400 secretory granules can fuse simultaneously, form this massive endocytic vacuoles, and after that it basically disconnects and start moving through the cytoplasm of the cell. So the bad news is that inside of this endocytic vacuole there is activation of the trypsin. So trypsin is a powerful protease. So this is something which is of course essential for the chemical digestion. This is something which will break proteins in the intestine. And in this case, trypsin is actually activated inside the cell. So this is 2 pancreatic acinar cells, and we are using not fluorescent, but fluorogenic molecules to detect this activation of the trypsin. They are not fluorescent until they're actually cleaved by the trypsin. So these fluorogenic molecules, activation, this appearance of the green fluorescence indicates that inside of this endocytic vacuole there is activation of trypsinogen, formation of active trypsin. Powerful protease is now, it appears inside our cells. And this could potentially result in the damage of the cell to autodigestion of the pancreas, which is observed in clinical acute pancreatitis. So this is basically just spectral analysis showing that we are indeed recording cleavage activation of these molecules known as BZPAR. So this is the sensor for the trypsin inside our endocytic vacuoles. So kind of it would be just sitting in the endocytic vacuoles, it probably will not be that dangerous. But the problem is these endocytic vacuoles undergo rupture, so they break, so the trypsin can actually leaks into the cytosol of the cell. So now all the proteins of the cells are accessible to the powerful protease, which actually is thrown from this endocytic vacuole. There is an interesting kind of properties of these organelles. They undergo actination. You can see that kind of this endocytic vacuoles is coated with actin. Some of them are coated, others are not coated or incompletely coated, fenestrated. And we think that this actin coating, so what we observe usually that kind of when vacuole rupture, it usually ruptures through the holes in this actin coat of the vacuole. So we concluded that actin is probably playing some kind of stabilizing role, trying to prevent the ruptures of endocytic vacuole. But kind of this mechanism is overwhelmed during pancreatitis. So this basically just shows the process of rupture of the endocytic vacuole through the actin on the surface of endocytic vacuole. Work which was done by kind of Misha Chvanov and Francesca De Fabere. These endocytic vacuoles can also undergo secondary exocytosis. You can see that one of these endocytic vacuoles already containing dextran, and after that we change the dextran and extracellular fluorescent molecules in extracellular solution, and you can see that this vacuole can fuse with the plasma membrane of the cell. So it can rupture inside of the cell, deliver trypsin into the cytosol of the cell, so that it can start chewing proteins inside the cell. It can also throw the proteins, it can throw trypsin into extracellular media, so that basically trypsin can now attack cells from outside. So this is kind of, we are moving to later part of the study. So there was significant body of evidence suggesting that autophagy or deficiency of autophagy is important for acute pancreatitis. And we decided to look at this. And we used animals expressing marker of autophagy and important proteins for the activation of autophagy. It's called LC3. So these animals express LC3 labeled with GFP in all cells of their body. So we can basically just isolate the cells of interest. In our particular case, it is of course pancreatic acinar cells, and look at autophagy in pancreatic acinar cells. So LC3 is green. So accumulation of the LC3, accumulation of the green fluorescence on the surface of organelle means that these organelles undergo autophagy. So these are autophagosomes. So again, a very simple experiment. We just isolate the cells, eliminate them, and just monitor how they generate autophagosomes. And this is how the autophagosomes are generated. I'll just play this movie again. So these are the autophagosomes generated in this cell type. When we look inside of these autophagosomes in condition when we have our fluorescent dextran and extracellular solution, what we observe that these autophagosomes are actually hybrid organelles. So they are simultaneously endocytic vacuoles. So our endocytic vacuoles undergo autophagy. So they basically precipitate generation of autophagosomes, coating of these organelles by LC3 GFP. This is astonishingly quick process. Normally, autophagosomes autophagy requires hours to complete the complex process of the formation of autophagosomes. In this particular case, complete autophagosomes are established within 15 minutes. So this was the first very unusual property of these organelles. The second surprising property of these organelles was that something which is normally used to visualize, to reveal autophagy. This is baphyllomycin. It is used to block progression of autophagosomes to autophagolysosomes, and used to accumulate autophagosomes. This, instead of the kind of creating more autophagosomes, completely blocks their generation. So Another toxin, conchylamycin, similar effect. So we are clearly dealing with a very peculiar form of autophagy. Next experiment showed that classical inhibitors of autophagy, MRT compounds, wortmannin, inhibitors of PI3 kinases, actually don't inhibit this very peculiar autophagy, which we are going to study in our cell type. And final surprising observation was that this actually autophagy is occur on a single membrane organelles. So this is experiment which is known as correlative light electron microscopy. So these are our cells expressing LC3 GFP. This is our dextran, which is accumulated in endocytic vacuole. So this is our endocytic vacuole coated with the LC3. So this is a hybrid organelle, endocytic vacuole, and autophagosome. We visualize this organelle in fluorescent microscopy, starting from live cell, and after that we fix these cells. Cells are maintained on a gridded cover slip. So we know location of the cell, and we know location of this organelle. And of course, we can also use confocal microscopy, so we know approximately the level of the Z slice at which this organelle was observed. After the cell is fixed, and we take it to our collaborators, laboratory of Ian Pryor, electron microscopy laboratory, and observe this organelle. So this is basically, we find the same organelle, which we visualize using light microscopy, fluorescent microscopy, and electron microscopy, and look at membrane organization of this organelle. And as you can see, this is a single membrane organelle. And this is actually a strong indication that this is non-canonical autophagy. And this is non-canonical autophagy of particular type. This is lab-like non-canonical autophagy. Lab is LC3-assisted phagocytosis. So this form of non-canonical autophagy was first identified in macrophages. And they kind of deal with the infectious macrophages, neutrophils. They have to deal with the viruses, they need to deal with bacteria, and they don't have time to use classical autophagy. In classical autophagy, organelle or structure is engulfed by double membrane autophagophore, which stretches around the area which need to be undergo autophagy, and slowly engulfs it, kind of closes, and after that kind of started to fuse with lysosomes and digestion starts. In this particular case, macrophage does not have time for this. Swallow dangerous virus, swallow dangerous bacteria, need to destroy it immediately. So in this case, phagosome is immediately covered with LC3, and this basically triggers fusion with the lysosome and destruction of the pathogen. And what is very peculiar, that the same process seems to occur in pancreatic acinar cell. So we have this single membrane, very fast, lab-like non-canonical autophagy. Very similar mechanism, actually. So this basically just shows that this is single membrane. This is the profile of this membrane. We, of course, get to visualize plenty of autophagosome showing that our electron microscopy is good enough to reveal classical autophagy, and there is no problem. But this is indeed single membrane organelle. So inside of this endocytic vacuoles, we can later observe presence of amylase, meaning that this endocytic vacuole was not able to release all digestive enzymes. Inside of this vacuole, we can observe presence of trypsin. In this case, we visualize it using antibodies against trypsin activating peptide. So this is actually something which is cleaved from the precursor of the trypsinogen. And it is a good indication that inside of this organelle, there is indeed activation of the trypsin. So this is correlation between the actination and LC3 conjugation of these organelles. So very elegant experiment. This was actually done by Francesca De Faveri and Misha Chvanov, showing that there is some kind of evolution of these organelles. They first undergo actination, coated with actin, and after that somehow actin is lost and they undergo LC3 conjugation. They form autophagy. The significance of these steps in the processing of the organelle is still being investigated. So this is kinetics of the, so LC3, the actination is very fast, and after that actin is lost on the surface of these organelles, and LC3, or in other words, autophagy, is actually develops. But just to remind you that everything, all these processes, formation of endocytic vacuoles, activation of trypsinogen, autophagy, actination, is actually downstream of the calcium response. So we can block these calcium responses using inhibitors of the RI1, STIM1 channels. Again, very efficient block. And this inhibition of the prolonged calcium responses drastically decreases the number of endocytic vacuoles. And this correlates well with the protective effect of this inhibitor of STIM and RI on experimental models of acute pancreatitis. So these are the results of 3 experimental models of acute pancreatitis. Serum amylase is the indicator of the severity of acute pancreatitis, and you can see that inhibition of calcium entry through STIM and RI strongly decreases the severity of acute pancreatitis. And this effect works well when the inhibition occurs before the induction of experimental acute pancreatitis. This actually inhibition works even when it was applied after the induction of acute pancreatitis. So 6 hours approximately, up to 6 hours after the acute pancreatitis is induced, these inhibitors are still capable of reducing severity of acute pancreatitis. So this again indicates importance of calcium signaling, calcium toxicity for this particular disease. So these are conclusions. So calcium releasing secretagogues produce local and global calcium signals in pancreatic acinar cells. So calcium signals are shaped by mitochondria and mediate efficient stimulus metabolism coupling in pancreatic acinar cells. Store-operated calcium entry in pancreatic cells, pancreatic acinar cells, is mediated by STIM1 and RI1 proteins. Calcium-releasing inducers of acute pancreatitis trigger formation of these large endocytic vacuoles and intravascular activation of trypsinogen. Store-operated calcium entry is important for these processes. And finally, these endocytic vacuoles undergo actination, non-canonical autophagy, rupture, and fusion with the plasma membrane. This is to say a few words about collaborators, about colleagues who have done most of the experiments for this study: Misha Chvanov, Francesca De Faveri, Svetlana Voronina, Mark Sherwood, Georgi Loshy Grishenko, Haley Dingsdale, and other colleagues involved in the study. And collaborators in prior electron microscopy. This is part of our unit. David Creedle, Robert Sutton, Ole Peterson, of course, was involved in many of the studies. So were Oleg Gerasimenko and Julia Gerasimenko. Okay, and these are the colleagues who supported our work with different valuable constructs. With this, thank you for your attention. And I will be, of course, happy to answer the questions.
Thank you, Alexei. The lecture was great, and I would like to attract your attention that the first slides were produced almost more than 20 years ago. It means that we span a time region of several dozens of years in this lecture, and definitely it is tremendous how many changes in the field have been produced, and now we understand everything much better than at the beginning of the research, when Alexei just entered the Department of Physiology, University of Liverpool. Okay, guys, do you have questions? I wrote you raise your hands if you would like to ask question, and I will give you this possibility. Okay, by this moment, no questions. In this case, I have a lot of questions and I will ask, but please also be prepared to ask your questions. First of all, you demonstrated many years ago that mitochondria are distributed in this very interesting way. There is this mitochondria belt or firewall around the secretory granule region, and there are other mitochondria on the periphery of the basal, nearby the basal membrane. But what is the reason for such a distribution? Calcium transients spread throughout the cell in several hundred of milliseconds, and ATP also diffuses in very prompt way in the cytosol. What is the reason to distribute mitochondria nearby places where actually, for example, calcium transients are produced? Anyway, they will be spreaded, and mitochondria at any site in the cell can feel this calcium.
I think that you probably know the answer better than me, Pasha. So I think that there is, of course, the concept of the mitochondrial calcium uniporter complex is, of course, is composed not only of the mitochondrial calcium uniporter, but also of the other associated proteins like MRA, and importantly, mitochondrial calcium uniporter regulators, MIQ1 and MIQ2. It's actually in neurons, it is slightly different. I think it is MIQ3 is also important for neurons specifically. But I think that these MIQ proteins are the guardians of the threshold of the calcium entry into the mitochondrial calcium uniporter complex. So the complex has a very peculiar property. It is kind of closed at a resting cytosolic calcium. And it is very important because otherwise this minus 60 millivolts mitochondrial voltage will continuously drive calcium from the cytosol into the mitochondria. And mitochondria, and consequently the cell, will need to pay significant amount of energy to pump out the calcium from mitochondria. So for this reason, the mitochondrial calcium uniporter complex guards itself against this futile calcium cycle by developing threshold, which is based on the calcium sensitivity of MIQ1 and MIQ2. So these are both EF hand calcium binding proteins. They detect calcium and they open the mitochondrial calcium uniporter only when calcium is above certain threshold. So there are basically genetic abnormalities of this MIQ1, MIQ2. So there are kind of mutations of the expression which actually manifest in this futile cycle. So people actually survive, but not particularly well. And futile cycle was actually directly demonstrated by the works of Michael Duchenne.
Okay, let's see, thank you. No, anyway.
So, so this is part of the answer, sorry. So this is because of this threshold. Mitochondria, which are far away from the calcium releasing channels, actually don't have enough calcium to move them above the threshold. So the kind of concentration which is necessary to open the mitochondria is in micromolar range usually. So if mitochondria sits very far from the calcium releasing or calcium entry channel, it simply cannot open mitochondrial calcium uniporter. So somehow mitochondria kind of try to be closer to the action of the calcium releasing channels. So this is kind of one possibility for the localization of the mitochondria.
Okay, no, it seems to me this is the main part of response. Yes, the main part, the main reason.
There is also a strange kind of almost theological kind of concept of mitochondria, which mitochondria can move to the areas which experience ATP depletion. So if there are kind of some ATP gradients throughout the cell, if there is a place for the particular kind of need for ATP generation, then mitochondria kind of move to this particular area somehow. I don't know, kind of. So this is something you can read in kind of some reviews and textbooks, but the specific mechanism is not particularly well defined.
Okay. Okay. And I also don't know, and you will increase my knowledge in the area, how this MCU dependent calcium increase in mitochondria is related in terms of signaling to changes in Krebs cycle.
So I think that the mechanism linking calcium entry into the matrix to the Krebs cycle is through dehydrogenases of the Krebs cycle. And there are kind of 3 limited calcium-dependent dehydrogenases, which actually mediate this response. The isocitrate dehydrogenase, oxoglutarate dehydrogenase, and pyruvate dehydrogenase are the dehydrogenases of the Krebs cycle, which is calcium-dependent. There are also studies by the Sarina Rizzuto and Guy Rutork's group, which shows that there is a somehow regulation of the ATP synthase by mitochondrial calcium. But primarily, kind of perhaps the most discussed mechanism, the most kind of established mechanism, is through the calcium-dependent dehydrogenases of the Krebs cycle.
Okay. And next question, how trypsin can be activated in vacuoles, because it is produced in pancreatic acinar cells as hematrypsin, and actually for its activation you need to use some proteases. What is the mechanism of activation in the vacuoles?
It can be auto-activated. So I think that what opinion of Miklos Sahintos, one of the main biochemists who worked in this area, is that kind of trypsin will always activate. So chymotrypsin, the kind of trypsinogens. So trypsinogens will always activate. So if you basically just leave it in the stationary in the vial, it will eventually auto-activate. So there are a lot of protective mechanisms. So there are trypsin inhibitors and secretory granules, but even they will not kind of last forever. So eventually, left unattended, trypsin will activate. Its status in zymogen granules is actually very, so it is kind of, so if it is just in solution, it will kind of, there is auto-activation of the trypsin. So I suspect that there is kind of relatively low activation energy of the reaction. So kind of from time to time, the cleavage occurs spontaneously. And after that, cleaved trypsin will rapidly activate other trypsin molecules. So trypsin can actually cleave trypsinogen and do this reaction. In the secretory granules, the precursors of the proteases, the zymogens, are actually not only in an active state of zymogens, trypsinogens, chymotrypsinogens, they're also osmotically inactive. So they are packaged in the kind of in the ball. So the mechanism of packaging is kind of rather complicated, but also involves calcium actually. So and kind of until, of course, they are in the fluid state, they are not particularly good at activating one another. However, when it is the endocytic vacuole, this initial kind of structure is condensed form of this proteases, precursors of the proteases, is diluted by the solution which is coming from the extracellular media. So in the endocytic vacuole, so this is not the condensed state of these proteases. So they are in fluid now, they can now start auto-activating. So this is our explanation. So not because 400 secretory granules fused simultaneously to form the endocytic vacuole, it has a very tiny fusion pore. Fusion pore is actually, can only pass the molecules with molecular weight of 70 K. So this is a really, this is not the flattening of the secretory granules on the plasma membrane. This is fusion through the very, very delicate fusion pore. But it is enough, of course, to let solutes to get water into the endocytic vacuoles. It disconnects and now unfolds. There is a this bouillon of the precursors of the proteases. They auto-activate. This is probably how it happens.
Yes, but it looks that vacuoles are composed of endocytic, no, actually secretory granules after endocytosis. Yes, and in this case, the trypsin should be expelled from the granules to the extracellular solution, and in this case, when you combine many endocytic vesicles and large vacuoles, in this case, there are no trypsin for.
Yes, this is yes. I think that this is probably what happened in physiological conditions when there are 2-3 secretory granules fused. This is actually work of Haruo Kasai, who is also another distinguished neuroscientist who worked in pancreatic acinar cells. If you fuse just 2-3 secretory granules, the life of the fusion pore is long enough to release all the trypsin, precursors of the trypsin, into extracellular solution by diffusion. When you fuse 400 secretory granules, and of course the last granule is very far away in this case from the fusion pore. And fusion pore only lives for a few minutes. There is simply not enough time to release all the precursors through this fusion pore into the extracellular solution. So it clearly retains amylase.
Okay, but can it happen in natural conditions? Because what you showed was done with application of, let's say, secretagogues for hundreds of seconds with high concentrations. Is it possible to wait that such a situation can emerge in pancreas in normal or pathological conditions?
This is difficult to say. This is, of course, very difficult to say. So this model of overstimulation is, of course, the model of a very peculiar form of pancreatitis, which can be triggered by phosphoorganic toxins, which can suppress inactivation of sodium channels in peripheral nervous system and upregulate release of acetylcholine. And this is also a model of pancreatitis, which is triggered by scorpion bite. So scorpion bite can actually trigger the over secretion of acetylcholine and this form of acute pancreatitis. So yes, in the very peculiar pathophysiological conditions, this could happen. It could potentially also happen in biliary acute pancreatitis when the outflow from the pancreas and gallbladder is actually blocked, and there is a potential of the reflux of bile into the pancreas. So bile acids, particularly taurolitoholate, is a powerful inducer of calcium signals in this cell type. So you can generate these large calcium responses in the cell. Actually, Mark Sherwood done experiment with perfused pancreas, stimulated with high doses of agonist, and observed formation of endocytic vacuoles. And Misha Chuanov also done experiments with the infused into pancreatic duct dextran, accompanied by the bile acid, and observed formation of balls, endocytic vacuoles, and giant autophagosomes. So as a model, it works. Of course, it would be very difficult to see whether this is something which will happen in clinical acute pancreatitis. We of course done it in human acinar cells, and the vacuoles form, and the basic mechanisms works as we see it.
I see, thank you. And maybe the last question is about the nature of other autophagosomes which are not produced from vacuoles. What is the difference, and what is actually the mechanism of their emergence in the pancreatic acinar cells, and what is the physiological function of this other small, let's say, autophagosomes?
Well, some of them can be all right and reasonably large, but probably not as large as this very large autophagosomes. phagocytomes, but some of them, of course, can engulf mitochondria and can be a few micrometers in diameter. So there can be. So we, of course, start. We done all the experiments with normal starvation of the pancreas. So there is of course, starvation will trigger normal autophagy. Autophagy is always present because pancreatic acinar cells are relatively long-lived, so they need to digest their and reutilize their organelles, and they do it by normal autophagy. It seems that there is also activation of classical autophagy, which, however, unlike this very fast lab-like non-canonical autophagy, takes approximately 4 hours. So 4 hours after the CCK stimulation, there is a development of the much slower classical autophagy. So this is possibly related to the damage of the cellular organelles, damage of mitochondria, for example, and classical autophagy, classical mitophagy of these organelles. But the processes are substantially different time-wise.
Okay, Alexei, thank you. I have some other questions, but I think that you are tired of my questions. Okay, I will check. Maybe someone else want to ask. No, you see, Alexei, in spite of quite clear presentation, the most part of our students, it seems to me, are involved in neuroscience research mainly, and I think this is why this area is not very common for them. Actually,
I think that the pancreatic acinar cells is basically just a cellular. It is cell biology. So the same principle of the stimulus metabolism coupling is working in a perfectly similar way in neurons and in cardiomyocytes. So the same type of the dehydrogenases were demonstrated. The NADH, FAD responses measured in mitochondria, classical works of Michael Duchenne, both in cardiomyocytes and neurons. So the trypsinogen activation is, of course, slightly peculiar. But I guess STIM-ORI also operate in neurons, and junctional complexes and calcium-dependent formation of the junctional complexes based on extended synaptotagmins was shown in nerve cells. So there is of course, it is basically just a general cell biology of the.
Actually, we had a lecture last week in which we precisely considered functioning of RI2 in terminals of Purkinje neurons and pyramidal neurons of the hippocampus. It means that the systems are quite similar, definitely.
Well, I think that there is RI1, both RI2 is present in acinar cells, although it is present in lower concentration in mouse, but higher concentration in rat. So in rat, it's actually RI2 is more prevalent than RI1, which is quite peculiar.
Alexei, and I can see one question in chat.
Yes.
Is any practical advice to prevent acute pancreatitis by inhibiting calcium entries through ORI channels?
This is a very good point. So it is now in clinical trials. So it is not the one which I showed you, but there are inhibitors of the STIM-ORI interaction, inhibitors of the store-operated calcium entry, which is currently in the stage 2 of the clinical trials.
Okay, and the second question, which kinds of processes connected with calcium release are connected with local and global signals? I don't understand quite well the question, but maybe you understand.
This is a good point. So I think that local calcium responses are
to physiological relevance of local and global calcium transients in pancreatic acinar cells.
So I think that local calcium signals are probably, so I think that when you stimulate with physiological dose of CCK and ACH, you probably remember this, Pasha, actually, because you've done some good work on this.
Yeah.
So you remember that the local calcium signals would drive secretion of the secretory granules. You have seen kind of small calcium transients coming out of, and kind of global calcium elevation and extracellular solution coming out of the, as a result of this local calcium signal. So local calcium signals are very good for triggering exocytotic secretion and triggering fluid secretion. Specifically, they occur in apical part of the cell. This is economical way of signaling. Global calcium responses, I think that calcium is also involved in the regulation of protein secretion, in growth, regulation of growth, particularly adaptive growth of pancreatic acinar cells, pancreas as a gland. So probably global calcium responses are more kind of something which need to synchronize the rest of the physiology of the cell with the secretion.
Okay, okay. As I can see, there are no more questions. In this case, Alexei, once again, thank you very much for your participation. And I hope other guys enjoyed your lecture, and it was, how to say, for me, it was some return to my past years. Thank you very much. You, how to say, you improve my knowledge in physiology of pancreatic acinar cells, and biophysics was just perfect. And I think that biophysical methods which you used in your lab can be applied to what we do here in our neuroscience research.
Wonderful. So kind of this is just the last moving. So this is the migration of the ERPM junctions in migrating cancer cells. And you can see that. So this is another interest of our laboratory. So this is junctional complexes, again, illuminated by GFP, by steam, by fluorescent steam. And you can see that this calcium signaling platforms, this fluorescently labeled steam, is actually localized at the leading edge of migrating cancer cells. So this is something which kind of just will leave you with this kind of last slide as a something which reflects other interests of my laboratory and maybe something to talk about in a couple of years.
Okay, okay. I hope not in a couple of years, but maybe next year. We will be waiting for your lecture devoted to migration, for dependence of migration of cancer cells on calcium signaling due to RI and steam. Okay, Alexei. Thank you very much once again.
Okay, all the best.